So today I will be describing the findings of Nirenberg, Matthaei, and Lieder. So once it was determined that 3 letters coded for an amino acid, the next big question was which 3 letters code for which amino acids. So the first experiment was done by Nirenberg and Matthaei. Their process was taking a string of U's and taking all the components out of a cell, which is called cell-free extract, and putting all that into a tube, and then adding all of the amino acids, except one was going to be radioactive. And then they did this same experiment 20 times, each time with a different radioactive amino acid, to see whether or not the radioactive amino acid was incorporated into the string of amino acids. They found that when the tube for phenylalanine was radioactive, it formed a protein. But phenylalanine was the only one that worked. So basically, they found that UUU codes for phenylalanine, and they also identified 3 other codons out of 64. CCC coded for proline, AAA coded for lysine, and GGG did not code for anything. Nirenberg also did another experiment with the help from Lieder. They took short mRNA sequences that had different codon combinations and added it to the mixture of ribosomes and tRNA, and all of that was attached to amino acids, and they did this over and over with different radioactive amino acids to see if that particular radioactive amino acid would bind to the ribosome and codon and tRNA. And if all of those were bound together, then they would be caught by the filter paper. They used this method to determine the other 61 codons. So these are the experiments that helped crack the genetic code. Thank you.